Okay. Does it work? So, okay, then I will just start with my presentation about collecting environmental DNA from thin air. So, usually if I give this kind of talk, I have to explain the definition of eDNA, but I guess in this round it's not really necessary anymore. So I can go over and save some time. Then, of course, this animation is still remaining there. But anyway, going to the question of how we can at the end collect switching now instead of collecting soil or water samples, going to collecting air samples is the question why we should do this. This is basically a third part of metabarcoding, and the solution or the answer of this is basically to detect, as usually, hidden species. As we already saw or listened from Oleg, there are still some situations where we find some fruiting bodies. The soil metabarcoding tells you there's nothing. So therefore, we have to handle this kind of absent information with care. And of course, we can try here air sampling and at least detect, hopefully here, some kind this species in the air itself. Of course, the same situation goes or will help for extreme or heterogeneous environment where it's quite complicated to find different species hiding sometimes behind ravines or something like this. And here the interesting part that comes now into the play is this time component. With air sampling, we are able actually to look into some kind of seasonality of spore release, define when our species, on species level, as you can see, on when our species releases the spore, give us the right timing to go out, especially if we do this opportunistic sampling. And in the past, we try to predict the weather and the snow melt events, and then go out. With some studies of spore release with air sampling, we are more or less able to pinpoint this time frame a little bit better in the future. And with all this data, of course, we are then able to make this kind of defining the direction of gene flow of our communities, described in a more understandable way, especially the community assembly and why they are assembled in this direction and not in the other way. If we look at comparing now the different metabarcoding areas, soil, water, and air, the big three, so to say, there are a little bit more. And look, what can we actually collect from our myxomycetes? Where is the DNA coming from? You see in soil and water, they don't differentiate that much and give us exactly the problem that when we find some DNA there, we don't know which life stage it was. Was it from a spore? Was it from the myxoamoeba or from the plasmodia or any kind of other stage? For air, it totally looks different. We have now here nearly exclusively our spores. The plasmodia and the myxoamoeba, as nature tells, should be on the ground. They don't fly, as far as I know. So therefore, we don't collect them, we don't expect them in our air samples. As you as well can see, well, why do I point on my screen? There are still remaining some unknown variables, myxocyst and sclerotium are expected as well on the ground, on the soil, but due to some accidents, circumstances, of course, these small particles can learn to fly, can be airborne and fly a little bit some distances just because of their small sizes. And therefore, certain particles can fly a few meters and then can be collected. How can we collect now these eDNA specifically for air samples? In most cases, it's super easy. Just through a mesh, for example. It doesn't need to be this metal mesh. The mesh size is a little bit big in this case, but tissues is already enough. Of course, we can wash out with liquids in a cyclone way. The same goes actually with an interplay with different air pressure and inertia of particles. Basically the same as we do it with liquid systems, but just without the liquid. And we can, of course, use the fact that our spores over time get an electric load on them, and we can actually sample them with this electric load on this electrostatic field. The further steps sometimes determine what we can do with the samples. If we look just on the quantifying elements, saying, is there this species in our sample or not? Basically telling us we have a PCR included in the steps, then the filter type is quite important that we use. So the liquid system uses certain kind of liquids. Can you just transfer it to the PCR or not? Cross-contamination is not solely critical for if PCR is involved, but is at the end one of the major critical points that need to be attained to. And this is the switch of the isokinetic sampling. If you have to take care for that, this is the difference between you do a quantitative or a qualitative analysis of your air sample. Quantitative, in this case, you want to know how many spores, for instance, you collected, and therefore the density of air in this certain time front. Of course, I can't expect that you know what isokinetic sampling is. I prepared more complicated an animation that tries to explain you what is isokinetic sampling. So it's always the relation between the environment, the wind velocity to the collecting velocity. So if both velocities are more or less the same on this side, then the representative amount of spores that you collected in this time frame represented quite nicely actually the amount of spores that are flying around in the amount of volume that you collected. If you collect faster than your environment or slower, then you start to make some kind of strange collecting, separating your entire situation so that you collect, for instance, critical always the small particles where they have a low inertia forces. They will be highly influenced by your different collecting speed, and therefore you can't tell anymore that the amount of spore is representative to the environment at this time that you collected. So enough of this explanation. Going to what kind of different air collector systems exists. There are majorly two groups, the active and the passive ones. The active ones, most common is the cyclone sampler. These devices are quite big and therefore expensive at the same time, even though that the principle is quite easy to do. Then, of course, there are different types that you can use. I just go them through to save some time. The electrostatic precipitation system exists as well, if you're interested in this kind of stuff. And the passive ones. The passive ones, as you can tell, don't need any kind of energy, but have the problem that the efficiency of sampling something is quite low. Other advantages is actually you can set them out in your environment and you can stay there for basically ever. Just have to take care that your sample is not degrading over time. The thing that is, the idea that was now coming up was, what is if we can build up a device that is active, is basically efficient in collecting something, but at the same time does not have this disadvantage of this active, all the other active samplers of being super expensive, super huge, and is not really applicable to non-institutional researchers. And coming up, hopefully, this is the other one, with our RDNA collector, right now in the prototype situation of the second generation, and it works as you would guess. On one end, wind comes in, on the other end, it comes out, going through a filter, and hopefully all your samples will be collected on some kind of tissue. As you can see, most of these elements are 3D printed, so it's more or less easy to rebuild this entire device. For non-institutional researcher, you just have to find an 3D printer or any company shop that does this. So every device is more or less in the center area, except, as you can guess, probably the van. That is the major motor driver that basically gives the entire principle. This is the most expensive one. It needs to be some kind of special one that generates a high pressure. So the entire device ranges therefore for around 40 to 50 euros per device. So the major part is coming from this motor. As you can see, strangely, it uses magnets, silicon gasket. It's designed by that you can easily switch the filter system in less of a second. It attached to itself so that I tried to minimize the contamination risk by switching the filter system, the filter caskets. Then, of course, a positive control if this device works at all. 15 centimeters apart from this device, I placed a matchbox full of myxos, tapped slightly on the button and see if you can collect. I saw nothing, but apparently there were something. Then we started already with the first experiments to further validate how well this device works or how unwell. And I will just give you a small glimpse of one of the experiments, the time experiment. As you can see, we used for the institution quite normal tissues. Most certainly you can use basically any tissue that you can find. The only critical part is if it's too thick, then you need a stronger blower or a stronger van, but that way you use already is quite strong. As you can already see, the motor is 12 volt, so there's a little bit of power behind it. And I used the opportunity last time when we went to our Transact to collect some data from the environment. Later on, Mahu, after me, will tell us something about this Transact that we visited already a few times over many, many years. So that will be interesting. And of course, some visual impression how the device is installed or set up in the environment. So now it explains why it has this funny little middle ring stuff. So you can attach actually on twigs. Of course, you can just take with you a stick and push it into the ground and then attach the device. Right now, as a prototype typical, it has a super nice switch. Doesn't have the risk of cutting yourself right now. And the battery is not visible. It's actually oversized. The battery is still working over like 20 hours collecting time. So far, so good. And now the some results of this collecting. As you can see, the different time points, 10 minutes up to 2 hours in sequence, because I have only one device, so I can't put them on the same time, collecting the same spot at the same time. Here are some pictures. We will look later on more in detail. I tried my best to look into the microscope and make some visually confirmation if there are some spores. It's more or less a little bit limited because you can't magnify it that hard because it's an open droplet and I don't want to collect the sample on the objective. And the funny thing is the 90 minutes visually was clean of mixers. 30 minutes I messed up. The sample is safe, but I forgot to look into it. And then, of course, what is the fun in just looking at the sample and see if there may be some mixers or mixer-like spores? I did an PCR with one of our SSU markers. If we can even find some gene markers or some mixes confirm basically what I saw. And you see already the results. Interestingly, where there was nothing to see, there is nothing. Then the 60 minutes, I don't know what happened there. Bad crushing of spores, maybe. And interestingly is that we have, even in our 10 minutes, in our short sampling time, found something. So we go a little bit further into this 3, starting by the 10 minutes. Again, no high magnification on this part. If you look really hard, you find some of the spores. A quick measurement tells us it's in the ballpark of myxos. Trying to magnify it with the computer as much as possible. Going to the 90 minutes where somehow we find nothing. And oops. With a sophisticated search algorithm, nothing was to be found. And now just look into the huge mess that we can create. This is basically this one. What you can see right now is huge amount of resin particles that learn to fly, huge amount of insect cuticula. There are some legs even there. If you go further up here, there is an entire head of some wasps. Then you have here is the collection, the beginning of the clump of filter fibers. So this is an issue right now, and so on and so forth. But again, if you look hard enough, you find again mixes. Even one is broken. Measuring them is interesting that we find them in different ranges. So indicating the possibility that we have even different myxospecies. Exactly. And what you can do further on is, of course, to make some kind of back trajectories to assume where possible your spores that you collected at this time came from. Of course, this is right now a first attempt to do this. Most of the cases you need to do some kind of dispersal kernel to say, okay, the chance of that your spores came from far away is in this proportion, while most of the cases, the perpetrator of creating the spores is basically right next to you. But as you can see, with this playing around with the high split model and going back 72 hours, you can say, yeah, some of them have their origin in the Polish area. And some of them are close by. The green dot is where the collection started, basically. Of course. So then somehow there are some major challenges that remains to be handled for this device itself. One of the biggest one is basically for all metabarcoding experiments is this contamination risks, especially depending on what kind of methods you use to sample the air. Generates different problems, especially if you use liquid one. Filter one, for instance, has the problem that as soon as you touch something or at the same time while the device is collecting, you start to collect, it's not wise to collect right in front of the device. You can do it, generate basically positive results, but where's the fun in it? And of course, if you do long-term collecting, you have the issue of how to store the stuff, the filter, so that it's not degrading before you recollect your stuff, your filters, or the liquid where it was stored. Then we have maybe, not always, sometimes you don't care for separating all the remains, the residue that you are not interested in, but if you care for, there is, of course, this lab-on-a-chip approach that I want to establish to separate, filter out certain kind of condition, dirt particles, or even separate certain sizes of spores. It would be interesting to use this for the parasite that we find now in the myxos. if this is possible, because this lab on a chip can be hooked up on a microscope, so there's potential to use this in this kind of. And with the last remaining part is the sensitivity. How sensitive is the entire system, especially when we find only a few spores, are we able to detect these few spores? Point of time investment, I think that is more or less clear that we have here the situation. If you sample longer, then of course you collect more and you will find more. Sensitivity point is I tried a little bit, played around with our SSU markers and reduced the number of spores that we crushed and then tried to amplify them. And we can go down quite good with 5 spores. So we just need 5 spores so far to detect a species. 3 spores would be probably possible as well, but somehow I managed to get a contamination in there. And single spores seems to have some problems still remaining. This is actually for the single spores, the second repetition, and it's basically always the same result. One is quite clear, one is completely out, and the third one is somewhat. So just to be on the safe side, 5 spores is doable for determining one species. Then with the final slides of asking what is the purpose for, is it actually a rock relief or does it more work? Comparing to with soil metabarcoding, it's actually quite similar. The minimal additional time investment is you just have to set it up, switch the filter, and that's basically it. The device itself is lightweighted. It's this size, nothing more. It's simple to operate it right now. It's just a switch on and off. Maybe in the future I will implement so that you can determine the time, how long it should run. The existing metabarcoding protocol doesn't need to be really modified on this part. Just at the beginning, how to get the stuff out of the filter. Super easy. Right now we vortex the entire filters and then we have it. And of course, the entire field of air sampling is quite new. Therefore, the turnover rate of new protocols, new changes are quite high. So therefore, you can assume they are still changing over time. And sometimes it's annoying to adapt. Yeah. And with this slide, I will finish my talk about future plans with this device, optimizing the entire device, especially getting out the contamination risk as much as possible and looking into this lab on a chip approach so that we have a whole system running around this air sampling system. Yeah. Thank you so much so far.
Thank you. It was a very interesting talk indeed. Do you have any questions?
In this experiment, when you tested 10, 5, 3, and 1 spore, how many species you tested?
Only
it is only one.
Only one.
So it can be different for different species, maybe.
Most likely. Yeah. Mm-hmm. It depends. My assumption, especially for the single spores, is because as you go down with the spores, that the risk of you use a spore that is actually not working anymore in this kind is totally degraded is quite high. Therefore, there are quite a lot of components that need to be looked at and say, okay, 5 or 10 spores is feasible, gives you a high chance that you detect something, but it's just interesting too, how far can you go down to still detect something.
And what was the species?
I forgot. Angela, Meryderma. Some Meryderma.
Yes, very nice talk. And I'm always very, very fascinated as when we as researchers are building our own sampling devices and so on. So I can very strongly relate to this. Also, very nice also Umsetzung. So you did it very well, I guess. I'm an electronics engineer by my history, so I would like to discuss it afterwards a little bit. There pop up a lot of ideas immediately. Air as sampling medium is, I think, very, very interesting also for a lot of you. I just wanted to mention, you showed in the first or second, in one of the first slides, this thing again, I have to mention it again, sporocarbs in soil. So when I do my experiments and I use large, high soil columns, okay, and with pure porous soils where you have large pores, it is not as these are not the single observations what I make, so it comes up quite often with certain species, for example, Didymum squamulosum. If one of the major factors for the sporocarp inductions, which I know for squamulosum, namely that the food source is not available anymore, so they are running out of food, they even sporulate in 5 centimeter deep soil columns.